Hey everybody. First of all, thank you for your interest in my talk. My name is Josh Auld. I'm a faculty member at Westchester University. I want to tell you a little bit about some of the research I'm doing there with collaborator Allison Kolpas. She and I received a grant from NSF. Some of this work is supported by that grant. And also just want to acknowledge our student co-authors, Ryan, Jeremy, and Chris. They all contributed to this work and they've all gone on to grad school at this point. So research in my lab is really focused on understanding the evolution of life history expression, mating system expression and evolution, and how this happens under variable environmental conditions. And predation risk is one of the major environmental factors that I'm really interested in understanding how it affects these traits, right? We know predation can have selective effects on the evolution of life history traits, for example. Think of Resnick's work and how a population can evolve to specialize within a particular predation regime in a way that maximizes fitness. Well, predators can also have inductive effects, right? Where individuals detect the presence of a predator and alter their traits, perhaps in an adaptive way, but in a plastic way that might increase survival in that environment. I'm really interested in understanding how those inductive effects can affect the expression of life history early in the life as well as late in the life. So I've done some work thinking about how predation risk can affect when you start reproducing. Today, I want to talk about really how long an individual reproduces and how frequently they reproduce in the presence of predator cues. And talk about this in the context of shifting resource allocation among different pots, growth, defense, and reproduction, and how organisms can maximize their allocation strategy. So I work with a model system called Physa acuta. It's a common freshwater snail, simultaneously hermaphroditic, prefers to outcross, but can self-fertilize. When it mates, it can store a whole bunch of sperm that it can use for months on end to lay outcross eggs. It's a really great system to work with. It's worldwide distribution, short life history. So it makes it conducive to studying the whole life history within a particular semester even, or academic year, for example. So it's conducive to working with students. We know from previous work that's with this species that it can express a wider range of inducible defenses, morphological traits, behavioral traits, and some life history traits. But I'm gonna try to explore that a little bit further today. So to build a framework for how I'm thinking about this, the framework for thinking about the life history, think of an individual's life history as a line where there's birth and death at the beginning and end of that line. At some point, the individual becomes mature and shortly thereafter should start reproducing. After that, they should continue to reproduce until eventually they die, right? The gradual deterioration of our fitness, the increase in death with age, senescence, is a known phenomenon and occurs in this species too. I'm showing here where the reproductive events are actually becoming more spread out. That's a hypothesis I want to test here. But what we have observed is that individuals actually tend to live beyond their last reproductive event, a post-reproductive period. Now, my hypothesis for that is that it is really just an ever-increasing reproductive interval during which they die. Right? So the reproductive interval is a focal trait that I'm interested in for this work, and it is the time duration between successive reproductive events. So we're going to follow individuals throughout their whole life and track how often they reproduce and use this reproductive interval as a comparison point across treatments. Really, I'm interested to see how does predation risk affect the timing of these events and the duration of the reproductive lifespan. So we do this experiment by collecting individuals from a wild population. I call those generation 0, bringing those into the lab and letting them lay eggs. We rear their offspring in cups like you see here on the left under standardized density and food availability. We rear up that generation 1 and use individuals from generation 1 from different generation 1 families as the parents for generation 2, where generation 2 is going to be the focal population. So when these generation 2 snails start into their treatments, when they're mature, we group mated them, individually identifying them so we can put them back into their treatments. And we're following the individuals throughout their entire lifespan. We do water changes on these tiny little boxes. If you can see the boxes in the middle picture, we do water changes 3 times a week on Monday, Wednesday, Friday. And it's within that we can apply our predator treatments. So we manipulate predation risk by holding, in this case, crayfish in separate tanks and feeding them snails, taking the water that they were held in and adding that to the experimental snails, or clean carbon filtered tap water. Okay, so snails in the no predator treatment are always going to receive that clean tap water Monday, Wednesday, Friday. Snails in the always predator treatment, number 3 in this, are always going to receive predator Q treatment. We know this is an effective way to manipulate predation risk, but we also know these cues break down. They're only good for about 24 to 48 hours. In another treatment, which I'm going to call the 50% predation risk treatment, snails received alternating water, right? So in one given week, they may receive clean water on Monday and Friday and predator Q water on Wednesday, and then that reverses the next week. So this is sort of an intermediate predator treatment. Here are the results showing the age at first reproduction, last reproduction, and death across these 3 treatments shown here on the y-axis. See, there isn't really an effective predator cues on the age at first reproduction, but there is a significant effect of predator cues, both levels, on the age at last reproduction and the age at death. So predator-induced snails are stopping reproduction earlier and dying sooner, okay? This figure on the right is just to show you that they are bigger when they reproduce. So this is consistent with other experiments. They are getting bigger, they grow faster. This is consistent with a strategy that we've seen before. When they smell crayfish in the water, they get as big as they can as fast as possible. Interestingly, we're not seeing a delay in reproduction here, which we sometimes see in association with that early investment in growth. Here shows you the survival probability. Just focus on the solid lines, the survival probability across the 3 different treatments. So the blue one on the left is the no predator, the purple is 50%, and red is 100%. If you compare, for example, the 50% survival points there, you see that there's a significant reduction in the curve, a significant shift in the curve to the left with predator cues. And actually, when we use hazard ratios to compare those curves, we see that there's a significant difference between the predator-induced treatments and the no predator, where the predator-induced treatments are dying sooner. So that's just confirm what you saw in that previous graph. Despite that, total amount of reproduction is not actually all that different. Neither is clutch size. So the graph on the left is showing you the number of reproductive events and the number of eggs, just on 2 different axes. You can see they're effectively the same thing. No significant treatment effects here, and no significant treatment effects on clutch size. So the snails in the predator treatment are dying sooner, but they're not reproducing more in any given clutch, but they're laying about the same number of eggs, right? How's that happen? It's the reproductive interval. It's that time between successive reproductive events, and it's decreasing with each level of predation risk. So you can see in this graph, the reproductive interval goes down with predation risk. In addition, we can track how clutch size and reproductive interval change over age, just to illustrate that reproductive function is senescing. Here I'm showing these as averaged across the 3 predation treatments because there is no treatment effect on this. So in other words, the rate of senescence in reproductive function is not faster under predation risk. It's just that the whole life history is actually sped up. They're doing everything faster. Their clutches are about the same. They're just churning them out at a faster rate. Okay. So to sum that up, we know that predation risk has effects on the lifespan. We've seen that before. Here we're actually illustrating that it's also effects on the reproductive lifespan, and that that's happening by accelerating that whole process, by speeding up the reproduction itself, right? And I think that explains the existence of a post-reproductive period. If the reproductive interval is gradually increasing with age, there's a better chance of an individual dying in that reproductive interval before they reproduce again. So perhaps this post-reproductive period is actually just an outcome or an artifact of an ever-increasing reproductive interval. When we look at this in the big picture, we see that what's really happening is these snails are investing in a set of different fronts: defense, growth, and reproduction. And under predator cues, they're really burning the candle at all those different ends, investing all of their resources really on all those different fronts. And collectively, I think that's why they essentially burn out sooner. So a major question then that remains in my head that I would like to address in subsequent experiments is, how do these inductive effects actually play out in nature? What evidence do we have for them in nature? How do they tease in with all of the other predator cues that a species might experience in nature, and how does that change over time? Those are some questions that I'm working on. I'd be happy to talk about this further or take any questions. Thanks for your time.